Welcome back to AgriTalk. On today's AgriTalk, we're talking about plant biotechnology, and with me in studio is Professor Richard Dur, who is the Director of Research Support and Dissemination at the Kenyatta University. And we are talking about how we can use technology to produce more food to feed the growing population. Dr. Before we went on a break, you're telling us a little bit about how we can use biotech to produce enough food, even as a country. Of late, we've been talking about we've been pushing the government to find out, do we have enough food in our reservoirs to last us the whole year? And the reason is most farmers, especially as you know, as a country, we rely a lot on maize. And of late, the last few years, farmers have been complaining about the low prices of maize at the end of the season. So most of them have dropped growing maize. And even as we speak, we are competing with cows for the same maize. So the maize that ends up on the table is reducing by the day. How can we, and yet at the same time, the land that we have is also reducing. So what can we do to make sure that even with these small pieces of land that majority of our farmers who are small-scale farmers still produce enough maize for the population?
Thank you, Philip. I think that's a very good question because the arable land is fixed. Actually, it is diminishing depending on how the climate goes, and there is also competition for the maize that we develop or we produce. Biotechnology comes in handy because one, it can increase the yield. When you make maize that is drought tolerant, it means there will be higher yields. If you make currently cassava that is otherwise has cyanide in it, if you make it edible, then it means you're increasing the alternative sources of food. Then if you make these production constraints like striga, they have a problem, in how to reduce the production constraint. If you are able to make maize or sorghum or rice that are tolerant to striga, and this is happening at Kenyatta University, we have very able scientists, and I would even mention that we have a team of researchers, including Professor Steve Runo, who is working very heavily and extensively on matters striga. Very, very, very detailed, scientist. We have other scholars in the lab and somewhere else also in the country. We have a number of them from Calro. We have some of them from Rebecca, and also other universities. The fact that they don't have the facility does not mean that they don't do research in biotech. They do them, but in the facilities that are in the country. One of the three, like most of the university staff, actually do research at Kenyatta University because that's the only university with that lab. So in other words, the best way to look at increasing production is, first of all, how do we attack the production constraints so that they don't really affect the production or the yields of our plants. So we look at drought-tolerant plants. We look at the cyanogenic, cassava. We are looking about on diseases. We also can make, BT, like Bacillus thuringiensis maize, BT cotton, which is already out there, released to different farmers to try get to know how actually the yields are now happening in different places in this country. And of course, you can also attack the soil ability for them to make the plants be able to absorb a little actively the nutrients on the soil. So yes, biotechnology is the direction, is the way to go, and this is something that we need to, continuously discuss.
If biotechnology is really the way to go, why is it that our own leaders, because I think if you ask me, our leaders are the biggest problem when it comes to distribution of biotech. They go out to the public and they tell them things that are not even, they don't have anything to back whatever they are telling to them. But they tell them it's bad, you will grow horns, all those things. But from the research perspective, we've seen the benefits of it. We've seen them rolling out biotech cotton. But when it comes to maize, they still don't want to roll it out. What could be the reason?
Well, it's because I think the leaders need to be sensitized, and we have tried as Kenya University
I think they have been
Biotechnology.
sensitized. They know.
They might not, they might appear to know, or they might also just have a position, which could be business, it could be culture, it could be all sorts of reasons. But from where I sit, I think over time now, they are beginning to see the sense. Because now we are not talking about importation of GM stuff. We have them here. So produced by, funded by, Kenyan government and of course taxpayers. So it is time for them to start actually appreciating or getting a bit of benefit from this taxpayers' money. But most importantly, I also think, Philip, that it's because in this country we have never grown from time immemorial. We have never grown GM plants. Nothing is out there. So actually BT cotton is the first that is going to go out there. So people really are just doing, academic exercise of engages. That is why it is important, and I really pray that the BT cotton goes several other technologies that are in the pipeline that we can still adopt. But we have spent a lot of time. Imagine the first GM product was out in the market in 1996. We're talking about 20 something years. 1996. And up to now, this country has not actually even benefited from that transformation of GM product. We're actually dropping the GM transformation, this normal genetic engineering, to another phase now we are calling genome editing, which is not, again, now we are not exchanging genes. So technologies are coming, back to back, and we have not even benefited from that. We are now moving to genome editing without even benefiting from the other one. Now we are discussing genome editing. Before that, we will move to another one. So are we just a country or a continent of discussing technologies? If this thing was really so bad, and I keep telling people, and this is not necessarily talking about superiority of people, but the US, almost 90% of the soybean is genetically modified. And I want to say, do you think realistically that a government like the US government will allow its entire population to eat things that are raw, that will kill the population? So sometimes I think that we are made to believe that this technology is not good. And I don't know who spread this unfortunate position, such that we are forever beggars. And it's sad, but I'm beginning to think that it is either we are designed to continue begging so that we lack food all the time, so that we are always given funds for this. Because why would such a country allow? It's not only that. China, Australia, we are talking even EU. Of course, Britain is a bit in between there, but all apart from just human beings, most of the animal feeds are genetically modified. And they say that that is for animal feed. Then I wonder whether they throw away the beef after the animals have eaten them. So it's all confused. I think it is time as a country to just decide for ourselves and say, what are we supposed to do? And the country, in the Big Four agenda, I think this is nicely outlined. That's why food security is top. And since the Big Four started, by the way, we are getting a lot more funding for biotech, because many of my colleagues are getting funding, and myself, I've got a number of funding that are coming from the government, which means that there is something that is changing. So the leaders just need to be told, without emotions, that please, there's nothing new that actually we are making. All we are saying is that we want to increase the production. And then they think that BT cotton is, is not edible. And by the way, for the fiber is only probably 40%. 60% of the cotton seed is actually oil.
That's what I was to ask you. And then again, I have an example. The cotton, the BT cotton they introduced in, was it Burkina Faso? Why did it fail?
Well, you would call it failure, but that's not the language that I use. I just think that they rushed the technology to get it out there, because it never failed. It was just because of the linen, the fibers were not as long. Okay? And therefore, when you get this, and that's what we are doing in Kenya, when you get this germplasm, which is genetically modified, you can now back cross it into another germplasm that has longer. That is just basically, if you want, it's assumed that if you want tall kids and you are, a little down to earth, short, all right, then it means that you go for relatively taller so that you mix them so that you get something in between. Okay, so it means, what are you back crossing? So you are short, but you want to back cross on a tall one so that you get something in between. So if you have a short one, but remember, this germplasm, for example, you want somebody tall, but this short guy is the person who is tolerant to COVID, if I might just bring it on board. Then you would rather have people, short is not particularly very good in terms of, and I'm saying this with a lot of humility, it's not like have any stereotype on any person's height, because I'm equally short anyway. So my colleagues, what I'm just saying is that we can take advantage of these people in terms of what is it that they have. And if I leave the human example and just go to plants directly so that I don't sound insensitive, Philip, is to say, if you have a germplasm that is tolerant to insects, and then the other one that has longer, linen or fiber is susceptible to the disease or to the pest, then what do? You cross it back into that so that you have something that is a little tolerant to the disease or pest, but also gives you the output that you want. Actually, it did fail. What they are doing right now is to try to back cross it into the right germplasm. It will be back.
Okay.
It will be back.
Okay. Now let's move to gene editing. You mentioned it a little bit. What is it?
Well, it's nothing really new or different from what we currently have. What it only does is that now we can get into the genome and identify particular sections that we want to edit itself without necessarily, before I have been talking about, if you want, say, something that is doctoral, you pick a gene from another source and you put it, like vaccination, where you're picking some drug from somewhere and you're putting it into somebody and the guy becomes a little tolerant. Now, when you talk about genome editing, we are not, because for a long time people have been saying, oh, you see, gene transfer means that you can pick a gene from even a pig and put it into maize that we eat, and the faith people come in. You get that kind of argument, and they are justified, by the way. Then others will say, why would you give me, genes from snakes? Because down there I said that the amino acids are only 21, whether they are in frogs or they are in snake, they are the shared. So we don't care whether the amino acid is coming from snake or it's coming from frog or whatever. It's just normal. So what we do in genetic engineering and genome editing precisely is that now we've kept the snake with also others we go into that very, very genome of that target organism and we edit it. We have mechanisms of running through the entire genome and identifying the section that you want to edit. Editing means that sometimes, by the way, if you suffer from, if the plant, for example, now has the cyanide that I was talking about, instead of now trying to silence that genome, because the RNA silencing or the gene silencing requires a certain layer, you bring something from somewhere and put it in, and then people will say transgenic. That's where the trans comes. But now here we just go into the genome itself, identify, edit the same genome so that it doesn't produce that. That means you have not brought any gene in, and now somebody cannot say that you have mixed the genome. Because it even happens naturally, by the way, because mutation happens naturally without even any external insertion of the genome. So we have techniques that we have in the lab where we can identify and the precise position which you want to edit.
Now, earlier on you talked about ethics and how important ethics are when it comes to some of these things. And I remember I attended some training or some conference where the first time I heard about gene editing. And one professor was saying, if you find someone malicious, you can produce a monster using the same. Is that true? And he was giving a relations to what we watch in movies. So he was saying, we can now produce a real monster that we can use in our movies using this technology. Is that really possible?
Well, theoretically, yes, it is possible, and I'll explain why. You see, let's take an analogy of even a knife. We have knives in our kitchen, but you can use it to cut your sukuma wiki nicely and enjoy, but you can also use the same knife to stab someone. The moment you stab someone using a knife, there are regulations and rules that will subject you to. In the genome editing and genetic engineering, this is probably the most tightly regulated technology. And therefore, we have ethics and protocols that we follow. We have to submit every time the reports of what we are doing in the lab. And even before it goes out of that lab, remember, this thing cannot be done in open. It is done in a biosafety level 2, level 3, and many other high levels of laboratory where it is double glazed, so nothing leaves, by the way, the lab. So by the time it gets out, it will have undergone all the processes of testing. And that is why even when it goes out like genetically modified crops, we take them for what we are calling confined field trials, somewhere with a drone which is regulated and with security, just to ensure that, first of all, it doesn't leak out before we complete all the research that is required to be done. So yes, if somebody malicious, even when you are in the confined field trial and you have a security officer who is malicious, he can still pluck the cob and put in the pocket and go eat it at home. You see, that will be somebody to be subjected through the laws of this country and arrested because it's not acceptable. So all the technologies have their either side. But by the time we release it to go out of the confinement and out of the lab, then scientists must have undergone, through all the processes that are required so that we don't release anything to the environment that will harm the environment and even the human beings. Remember, we also live in this country, and these scientists are also here. So they are more scared. They are fathers. They are also, people's sons and daughters. So we want to live in this country, and therefore we want the best for the country.
Okay,
Yes.
that is true.
Yes.
And that reminds me of the COVID-19. Um, some people are saying it was released from a lab in China. So those are some of the issues and fears that people have out here. But again, when you compare biotechnology, GMO, and gene editing, when it comes to crop production, will they still have the same production, or one will do better?
By the way, it is still the addition. It's additional. The beauty with science is that it's incremental. The genetic engineering was good because you can pick the genes from whatever source and put it in, and it's fine. And it also doesn't mean that once you have made a transgenic crop, you cannot continue and do, gene editing. Actually, I would even suggest that's a better combination. That you have a genetically modified, and then you go ahead and you find that this, you might not even get the gene out there. You can come into the genome and still edit. They can be used interchangeably. The only beauty with the genome editing is that this complaint that has always been there, that you are picking genes from left, right, and center, you'll come up with a monster, or that we don't know. It's no longer here because there's no gene that is coming from anywhere. You are just doing targeted mutations or editing that are meant for, improving that organism's availability and existence and probably ability to survive the ongoing environmental constraints. So you can use both. So you might not really compare which one is which, because even as we are talking, not so many products produced through genome editings are out there. So we are yet to get to know in terms of productivity. But yes, I would even suggest that do genetic engineering and do genome editing for optimized outputs of those organisms. Yes.
Earlier on, you'd mentioned other countries that have rolled out some of these products. Where are we in Africa? And why is Africa lagging in all this?
Yes, good question again, Philip. Africa is struggling, and for the reasons that are now becoming clear, that we've wasted time all this while. In Africa, we talk about South Africa. Actually, 1998, they already rolled out. 1998, the product was out, the first one, 96, 2 years later, then they were out. They've been enjoying the GM since then, and they are also in Africa. Then, of course, there was Egypt sometimes, and then, when the revolution came, they slowed down a little bit. We have Burkina Faso that we talked about, but because of they were retooling the technology again. We have Kenya here. We have Sudan has rolled out actually BT cotton while we are still discussing. All that guy said is like, is American eating this thing? Is whatever eating a South African on it? Bring it, because and that's some of the thoughts that we want some people who are firebrands who can just make quick decisions. Here in Kenya, we've made a lot of progress, I must say, because even as we are talking, we have now BT cotton out there for farmers to try. We have also BT maize at advanced stage and national performance trial. That's just one stage, and then another stage of multi-locational, and then we are out there. Okay, so we are making progress as well. But the challenge has been largely, Philip, because of one, the conspiracies that have been there, and number two, I think As a country, for example, Kenya, we have never had these plants out there. Therefore, we are just doing academic discussions. Put it out there and let the farmers decide.
Okay. And do you think maybe the leadership that we have is a problem in that some know, but they decide not to speak out, or even the ones who are supposed to make decisions know, but they opt to just let it
If
go?
you ask me, I think as a country, we have a lot more of the requisite, you know, institutions that can regulate this. These are the things, because sometimes it's because of lack of knowledge. But there are 4 or 5 things that are critical, actually, Philip, before a country chooses to decide to actually commercialize some of these things. You ask yourself very basic question. Do we, first of all, have the capacity, human capacity, to produce this stuff or to develop? Yes, we do. We have all the courses of biotechnology that are funded by even HELB, which is government funding. So we have the human capacity. We have the laboratories. We have the regulator, which is very critical, the National Biosafety Authority, with competent, actually, personnel. Actually, the CEO is a professor of biochemistry, understands these things. We have even the appeals board. You see, if you don't agree with what NBA says or the decision they've made, you can still appeal as an individual. So we are covered as a country. So sometimes I think we just need to be a little to sensitize our leaders, particularly politicians. But I want to tell you, Philip, we have seen, because we've been here longer, we have seen a lot of improvement, and even scientists and politicians and leaders reaching out. I've sat in several meetings where they are discussing, even with the religious leaders, in terms of what is your position on this. Actually, the government right now is a bit keen on ensuring that the food security bit is actually, solved, or at least, secured. And for that reason, more scientists. I am an example. I have sat in several meetings with the government, leadership, to discuss what is this thing that we can get from this biotechnology and other related disciplines. And I can tell you that things are changing, and soon we are going to have these things rolled out, and it will be a good time for us.
Okay. As we wind up, maybe in less than a minute, in terms of our institution of higher learning, you've talked about what the Kenyatta University is doing. Do we have enough? Can you say we have enough funding for this kind of research in Kenya?
In terms of funding, biotechnology is finance intensive, because before you release it out there, you need to have done all sorts of experiments to make sure that it is good to go. But we must also say that the government is doing a little bit more than it used to do before, which is very encouraging. Probably all we need, and with the coming in of National Research Fund, which you probably know, is a government funding agency, which draws funding from the government. At least now we have seen seriousness in terms of now fundings, and they release periodic calls for particular research. But for biotech, I must say that we are increasingly getting more funding, and we feel that the government should do more, because it cannot only be for Kenyatta University. Of course, other people like even Seku also has researchers doing, and when they get to, remember, tissue culture is not regulated, but when it comes to genetic engineering, it is regulated, and you have to do that section in a lab that is accredited. And therefore, as a country, we have seen a lot more of also funding coming for biotech, and also more researchers taking up biotech research, which is something that I really want to applaud the government for, and to ask them for even more funding going forward. Yes.
Thank you very much, Prof, for sparing time to be with us today. We've learned a lot, especially when it comes to biotechnology and why it is important for us as a country to adopt some of these technologies. We'll still call you next time. But for now, for our viewers back at home, that is it from us today. Until next time, goodbye.